Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Liquid Biopsy in Oncology, Opportunities and Challenges. Hi, my name is Sonia Hunt, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's situated on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas, including oncology, cardiology, metabolic disease, endocrinology, central nervous system, and antiviral and anti-infective. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Mustafa Bahasi. He's the Director of Clinical Laboratory Sciences with Medpace. Dr. Bahasi is a research scientist with over 20 years of clinical and research laboratory experience. He is experienced in biomarker development. Mustafa moved to the Department of Cancer Biology at the University of Cincinnati and later became a faculty member. As an independent faculty, Mustafa developed a translational line of research where he worked closely with clinical oncologists to develop new companion diagnostics using cutting-edge genomic technologies. And now it's my pleasure to introduce you to Dr. Lion Gleish. He's the Senior Vice President, Medical Department at Medpace. Lion has been directing Medpace's oncology program since 2004. Lion has served as a medical monitor on multiple oncology studies at Medpace. He is responsible for the development of study-specific training materials, as well as the ongoing training for the clinical operations group. Lion spent more than 10 years as an academic investigator, during which he became a recognized expert in new molecular therapeutics. And finally, now it's my pleasure to introduce you to Dr. Christopher Huth. He's a Senior Clinical Trial Manager, Clinical Trial Management at Medpace. Christopher has nearly 10 years of clinical oncology operations, management, and monitoring experience at Medpace. His experience includes the management of large-scale global studies, as well as management of FIH-targeted agent trials. Christopher has served as global CTM on multiple Medpace targeted agent programs. His expertise has been used not only for the trials he manages directly, but also as a resource at Medpace for developing processes and operational excellence in targeted agent studies. Now, it's my pleasure to pass the mic and the controls over to Mustafa. So Mustafa, when you're ready, you may begin.
Thank you, Sonia, and welcome everyone, and thank you for joining the webinar. So, Sonia, am I on the right screen?
Yes, you are.
All right, thank you. As everybody knows, the field of liquid biopsy is a very active area of research, and I will try today to present a high overview of the field in about 35 minutes, but we will leave time for discussions at the end for our panelists to shed more light on the current status of liquid biopsies in clinical management. So for those of you who are still wondering what the liquid biopsy is, A liquid biopsy is a minimally invasive technology for detecting molecular signs of cancer and other diseases in body fluids such as blood, cerebrospinal fluid, CSF, or urine. These include circulating tumor cells, or CTCs, circulating tumor DNA, or ctDNA, and exosomes. Finding biomarkers for solid tumors was limited to tissue samples collected via a tissue biopsy. But now with CTCs, cell-free DNA, and exosomes, we have other options. So for CTCs, these are cancer cells that split away from a tumor and circulate in the bloodstream. CTCs can sometimes exit the bloodstream and enter other organs, growing into new metastatic tumors. This tumor shedding in the blood can be measured to determine whether a patient has cancer. And the more shedding found in the blood, the higher the likelihood of a patient having late-stage cancer. As we will get to it in a minute, you will see that these CTCs are mostly abundant in late-stage cancer and less abundant in early-stage cancer. For circulating tumor DNA, or cell-free DNA first, is DNA that is shed into circulation during cell death by apoptosis or necrosis. Most cell-free DNA in circulation actually doesn't come from the tumor, but it comes from other sources such as muscle cells, endothelial cells, leukocytes, etc. But a very tiny small fraction derives from circulating cancer cells and tumors in cancer patients. The cell-free DNA that comes from cancer cells is called circulating tumor DNA, or ctDNA. As tumors grow, cancer cells die and they are replaced by new ones, and the content of dead cells, including DNA, is released into the bloodstream. Like CTC, ctDNA can be measured in a patient samples to screen for cancer. ctDNA is known for its higher sensitivity to alternative biomarkers. And then for exosomes, exosomes are extracellular vesicles responsible for cell-to-cell communication and the transmission of diseases. Exosomes are particularly useful as a basis for liquid biopsy, as they are secreted by living cells rather than secreted during cell death, like we talked about for cell-free DNA. As a result, exosomes-based assays offer a more comprehensive real-time window into the patient's health and have higher sensitivity. They also offer logistical benefits because exosomes are highly stable and easy to obtain and distinguish, and that's one of the limitations for all circulating biomarkers. So in the field of liquid biopsy, as I mentioned earlier, it's a very active area of cancer research. A recent survey of the ongoing clinical trials using liquid biopsy in cancer showed that there are almost 3,000 clinical trials involving CTCs in clinicaltrials.gov, and over 1,600 using either cell-free DNA or ctDNA, and there are fewer studies using exosomes as a liquid biopsy. These clinical trials, at least the ones that have results available, they have shown promising results for the early detection of cancer and confirmed these biomarkers as a good tool for real-time monitoring of acquired therapy resistance, accurate disease progression surveillance, and the improvement of treatment. So all this would translate obviously into a better quality of life and extended progression-free survival and overall survival for cancer patients. One of the limiting factors that is hampering the use of liquid biopsy in the clinical management of cancer patients is the low abundance of these biomarkers in circulation. For instance, CTCs, there is between 0 and 10 CTCs per ml of blood in early stage cancer versus thousands of CTCs in metastatic carcinoma, for instance. But that would depend on several factors. So it varies depending on cancer type. I will later show some cancers such as glioma that shed very little circulating tumor DNA into circulation compared, for instance, to colorectal cancer or bladder cancer, and the method of isolation. There are different methods of isolation of CTC, and the method of isolation that is used can make a huge difference in terms of the number of CTCs that are enumerated. So separation based on epithelial surface markers, for instance, compared to separation based on size and morphology will make a big difference. The concentration of cell-free DNA in healthy subjects is between 0 and 100 nanograms of cell-free DNA per ml of blood, with an average of 30 nanograms per ml, whereas the concentration of cell-free DNA in the blood of cancer patients varies from 0 to 1,000 nanograms per ml, with an average of 100 nanograms per ml. That gives you an idea again about how there isn't much of this cell-free DNA in circulation, which requires to improve technologies for detection and for analysis. For exosomes, there are about 10 to the 8 to 10 to the 9 exosomes per ml in serum or plasma, but still a very tiny fraction originates from the tumor, and the method of separation also can make a big difference in the number of exosomes that are recovered. So this is a study I was referring to that looked at the number of patients that are, these are both all the studies that I'm going to be talking about now in this particular slide. They are looking at late stage cancer. So they're not looking at early stage cancer. But as you can see on the left, the fraction of patients with detectable circulating tumor DNA can be 100% of patients we're talking about bladder cancer or colorectal cancer or ovarian cancer. When we look at the glioma patients with GBM, for instance, less than 10% of them will have detectable circulating tumor DNA. Although GBM is a very advanced form of brain tumors, yet the number of circulating tumor DNA is detected in less than 10% of the patients. So that may be explained by the presence of the blood-brain barrier, but again, GBM usually at that stage, the blood-brain barrier is very compromised, and then the circulating tumor DNA should be released in circulation. So in that area, when working with blood, it's extremely hard to detect any circulating tumor DNA from GBM, with exception of some overexpressed mutations like EGFR, the V4 variant of EGFR in GBM patients. Sometimes you can detect it in plasma. If you're looking for mutations in IDH1 or IDH2, usually you have to use CSF for that purpose. Other tumors that shed very little DNA in circulation, tumor DNA in circulation, include also thyroid cancer. So when you look on the right for the quantification of mutant fragments this time, the same thing. Neuroblastoma, for instance, you can detect over between 100,000 and 1 million copies of mutated DNA in circulation, compared to GBM, where again you're detecting less than 10 copies of mutant DNA. So it depends on the stage of the cancer. Stage 4 being the one that has the most mutations that are in circulation, and stage 1 the one that has the least. And then it also depends on the type of cancer, with bladder cancer, for instance, colorectal cancer having the most, and then glioma and thyroid cancer having the least. So for the one thing that makes a big difference in terms of analyzing these biomarkers is the isolation techniques. So because CTCs are ultra rare events with numbers as low, like we mentioned, as 1 CTC in 10 to the 6 or 10 to the 7 leukocytes in 1 ml of blood, enrichment and investigation of CTCs has been extremely difficult. Several methods are currently used to isolate CTCs, but each one of these methods has its own challenges, limiting their clinical implementation. So the one that has been used since the first FDA approval for the cell search system is immunofluorescence staining of CTCs. So this is using epithelial or tumor cell surface markers such as epithelial cell adhesion molecule, EpCAM, and cytokeratin CK. And the first and only device, the cell search, which was approved by the FDA for CTC enumeration, used this concept. But when we know that cancer cells, when they leave the site, the primary tumor site, they undergo epithelial to mesenchymal transition, also known as EMT. So when they leave the tumor site and move into circulation, we can imagine that this device will only detect very few CTCs, the ones that still have those epithelial markers. The ones that have already started the transition to mesenchymal form will lose those markers and will not be detected. So indeed, several studies have shown that there are sometimes 30 to 300 times more CTCs in circulation than what cell search was able to detect. There is also the antibody targeting of white blood cells for removal. So this is pretty much a negative selection. You use a CD45 marker to remove the white blood cells from the sample and then leaving just CTCs. But again, this method of negative selection, which use antibodies to CD45 to remove normal leukocyte, this selection is usually not very efficient, and CTC population is usually high, very highly contaminated with leukocytes. Also, some CTCs express CD45 surface marker. Those will be also eliminated in this case. So there are a lot of limitations when it comes to methods of isolation, and those can make a huge difference when we are enumerating or isolating CTCs. A size-based filtration of CTCs can be also used, and it's based on the cell's morphology, but their heterogeneity is a major obstacle for CTC detection. Again, the size and morphology, it does, all the people who use size and morphology for CTC isolation, this is based on the fact that cancer cells are bigger than leukocytes, but again, some CTCs may be smaller, so you lose those. Some of them may have same morphology or leukocytes, and then you will end up isolating leukocytes instead of CTC. So several strategies that combine size, shape, and immunophenotype and profile are being investigated, but the multi-step cell preparation process of doing all these steps, trying to isolate cells based on shape and immunophenotyping and all that process leads to CTCs being damaged and fragmented, causing inaccurate detection and interpretation. For cell-free DNA, however, cell-free DNA is much easier. There are a number of kits that can be used to isolate cell-free DNA and instruments to measure its concentration, such as the Agent Tape Station, Nanodrop, etc. And there are newer chemistries for NGS library preparations and platforms for cell-free DNA sequencing. So as a result, as I will be showing later, cell-free DNA is the most advanced liquid biopsy biomarker in the clinic, with several FDA-approved assays and many more being considered for approval. For exosomes, exosomes have shown great advantage in liquid biopsy. First of all, because the presence of large amounts, like I mentioned earlier, almost 10 to the 9th particles per mil in biofluids contribute to relatively easy obtaining of vesicles. Second, the exosomes are secreted by living cells and inherent abundant biological information from their parental cells. Third, exosomes are innately stable because of their lipid bilayer. The high biological stability allow long-term storage of specimens for exosome isolation and detection, and exosomes are being mostly used for detection of secreting RNA and microRNAs. Multiple platforms have been developed to isolate exosomes using ultracentrifugation, immunoaffinity, and filtration, but there are still a number of issues related to purity and sensitivity, highlighting the need to develop an easy to operate, high sensitivity and high purity platform for exosome separation and detection. So these limitations have translated to fewer regulatory approvals for exosomes in liquid biopsy, as liquid biopsy for use in cancer. So, as I mentioned earlier, although we still have a long way to go until liquid biopsy is fully incorporated in cancer management, the 3 biomarkers that I discussed today have shown some utility in cancer management and in predicting outcomes. For instance, for CTCs, the cell search system has been approved by the FDA since 2004, and CTC count in whole blood is an independent predictor of progression-free survival and overall survival in a number of metastatic cancers like metastatic breast cancer, colorectal cancer, prostate cancer, And so it has been used in the clinic for a long time. But again, like I mentioned earlier, there is a lot of limitation to using this system because it only looks at epithelial markers at the surface of CTCs, while we know that CTCs, after they leave the primary site, they transition to mesenchymal, and then they lose those surface markers. We'll discuss, later some of these issues with isolated CTCs. For cell-free DNA, the use of cell-free DNA to detect mutant fragment in circulation has been approved for many indications, either for screening, for disease monitoring, or for prognosis. Several assays have been approved by the FDA, as we will discuss later. I checked last night, there were 22 cell-free based assays that have been approved by the FDA. And I'm sure there are some more that are approved by the European Medicines Agency in Europe, but at least in the US, there are quite a large number. I will show in a table, but it's going to be very few of them. There are 22 of them that have been approved by the FDA. So just an example here, I'm showing detection of activating EGFR mutations for non-small cell lung cancer. So these activating mutations usually are a good indicator that the patients will respond well to EGFR inhibitors, such as gefitinib or erlotinib. Detection of T790M mutation in patients that are undergoing treatment with EGFR therapy is always indicative of resistance to these EGFR inhibitors, and T790M mutation can be detected either in plasma or even in urine. Detection of BRCA1/2 mutations and ETM alterations in prostate cancer as a companion diagnostic for olaparib, for the PARP inhibitor. Olaparib also has been approved. And again, the list is long of the FDA-approved assays for use of cell-free DNA or circulating tumor DNA. For exosomes have also shown some usefulness in cancer screening, especially in detection of ARC fusions in exosomal RNA in lung cancer, detection of BRAF V600E mutation in lymphatic drainage-derived exosomes in melanoma. So for, again, we're talking about the regulatory approvals. In terms of FDA approvals, all three biomarkers have received some regulatory approvals, although as I mentioned earlier, circulating tumor DNA and cell-free DNA has been leading the number of approvals, especially after the development of new technologies with better sensitivities and specificity in the NGS space. CTCs received the first ever FDA approval for cell search, the CTC enumeration platform. That's to monitor patients with advanced metastatic breast, colon, prostate cancers based on the cancer's level and circulating tumor cells. Recently, just last August, actually, the FDA approved the Parsortix C1 clinical system, the first FDA cleared medical device for the capture and harvest of CTCs from metastatic breast cancer patient blood for subsequent analysis. Again, this platform is not for enumeration of CTCs like the cell search, but it's for capture and harvest of CTCs for potential downstream use for NGS or whatever application they are used for. They are not for enumeration. So for cell-free DNA, like I mentioned, has the most approvals, especially in the companion diagnostic space. As of last night, there were 22 FDA-approved cell-free DNA listed on FDA.gov website, starting with the Roche Cobas EGFR mutation. So that has been approved for multiple indications as a companion diagnostic for detection of EGFR mutations. The TerraScreen PIK3CA for breast cancer, also in non-small cell lung cancer for EGFR mutations and for PIK3C3 mutations. And also the leaders in this space now are Foundation Medicine and Guardant 360. They all have a large number of FDA-approved companion diagnostic assays for different indications. So, like I said, the list is much longer than what I'm showing here, but you can always go and check it on FDA.gov. They have a list that you can search whether the assay is plasma-based or if it's other liquid biopsy type of assay. But again, Foundation Medicine and Guardant 360, they have a large list of FDA-approved assays. So for exosomes, I mentioned there are in 2016 the ExoDx long ALK was approved for the isolation and analysis of exosomal RNA from blood samples, and this was mainly for the detection of the AML4 ALK mutations in non-small cell lung cancer. And then in 2019, the ExoDx prostate intelligence score was approved by the FDA. So this score would predict patients with PSA from 2 to 9 nanograms per milliliter likely to develop high-grade prostate cancer based on detection of a number of gene RNA in exosomes. So these are the two, approved assays that I was able to find. I'm not sure if there are any more, but as far as my research goes, those are the only two that I found for exosomes. So when we look at the overall workflows for circulating tumor cells, cell-free DNA, and exosomes, they all share the same limitations. And at the preclinical and the pre-analytical, analytical, and post-analytical stages, let's start with CTCs, for instance. So when we look at CTCs, with the sample collection and stabilization, that's the very first area of limitation. So the tubes that need to be used to collect blood that will be used for CTC enumeration or for collecting CTCs, there are a number of tubes like KTA-DTA tubes, but they provide only stabilization for up to 4 hours at ambient temperature. There are like cell save, which is the tube that's used for cell search. Those allow stabilization for up to 96 hours. Ambient Strack tubes, 7 days ambient, PAXgene tubes, 72 hours refrigerated, etc. So there are a number of tubes that are out there, but there isn't really any standardization of the process. So we don't know, the stabilization of these CTCs, for how long they can be during the shipping process, how long can we afford to have them, for instance, shipped ambient, etc. So there's a lot of research that needs to be done in that area. The other thing is the cell search system. It uses 7.5 ml of blood, and we all know that with very few CTCs in circulation, especially at early stage cancer, 7.5 ml may not be enough to detect or to isolate enough CTCs. And there are studies that show that 40 ml of blood, for instance, allows detection of more CTCs. But again, sometimes it's not feasible to collect that much blood. So there are different issues that happen at the sample collection, stabilization, the choice of the volume of blood that needs to be collected, the type of cancer that is being looked at, and then whether it's a cancer that shows more CTCs than other types of cancer. Like I showed earlier, GBM has almost no CTCs in circulation, even at stage 4. So all those things are things that need to be stabilized and established. And then there is the issue with isolation. Isolation, we talked about positive selection, like the cell search system, using EpCAM as a biomarker, as a marker for isolation. And we talked about the fact that those tumor cells are actually undergoing epithelial to mesenchymal transition, and then how those markers may be fading away when cells leave the primary tumor and then they go into circulation. Negative selection using CD45 marker, we also talked about the fact that this leads to impure CTC population, also getting rid of some CTCs that may express the CD45 marker. Size-based enrichment, we talked about this newly FDA-approved Parsotrix platform that is able to isolate CTCs based on size, but again, CTC heterogeneity also is a problem when isolating based on size or morphology. There are some platforms out there like Deep Array allows sorting and recovery of single cells, from a population of CTCs. You can isolate single cells, and then you can use those single cells whether to test for cancer-specific gene panels. So I'm moving to the detection step. So you can use either cancer-specific gene panels. There are a lot of gene panels out there for lung cancer, for prostate cancer, for breast cancer that can be used to specifically interrogate genes that may be mutated in that type of cancer. One can use RNA-seq, microarray analysis, whole exome sequencing, whole genome sequencing. But again, when we are talking about a single cell whole genome sequencing or a single cell RNA-seq, the variability that is from one cell to another is so big that one needs to sequence a dozen or a few dozen single cells in order to have data that has reproducible and repeatable and accurate results. And when we are talking about doing RNA-seq or whole genome sequencing on a single cell, obviously it's going to be very costly and it takes a very long time, which makes it very hard to implement in clinical use. So, and then there is data analysis and the interpretation. So data analysis, most of the pipelines for data analysis are based on the GATK best practice pipeline, and the interpretation of variants is usually based on the ACMG guidelines. So those the data analysis and interpretation, although there are still need to improve those bioinformatics tools to better analyze the data. They are mostly based on this GATK best practice pipeline. So the complexity of using CTCs, or using the analyzed data from CTCs, and I talked about how not so accurate and then not reproducible. And then if you are doing, for instance, whole genome sequencing, you're going to need to do whole genome amplification first, which will introduce a lot of duplicates that you need to get rid of. So those kind of made the FDA really worried about implementing any CTC-based assays or approving them. So none of the CTC-based genomic, transcriptomic, or proteomic assays have so far gained regulatory approval for clinical use. So for cell-free DNA, again, cell-free DNA is a little bit easier because we have, some established protocols to do so. One can use KT-UDTA tubes, but stabilization is only for up to an hour. So cell-free DNA is extremely labile, very unstable. Half-life, it's about 140 minutes, and then so one can either collecting KT-UDTA tubes, but has to process cell-free DNA isolation within an hour. Or you can use Streck tubes, for instance. They allow stabilization of your cell-free DNA for up to 14 days ambient, or Paxogene cell-free DNA tubes for 10 days ambient. And for isolation, that's where I think what makes the big difference when we talk about cell-free DNA versus CTCs is the isolation process is much easier for cell-free DNA. We have a number of kits like the MagMax cell-free DNA isolation kit from Thermo Fisher or QIAGEN Mini-Elute cell-free DNA kit from QIAGEN. Those allow good isolation of cell-free DNA. So, and then for detection, there are a number of cancer-specific gene panels. I will come back to talk about some of the ones that are either like personalized genes that panels that are built based on the somatic mutations that are detected in the primary tumor from the patient, or some personalized kits that are commercially available that one can use either in-house or have it done through some of the providers in the field. qPCR and digital PCR can also be used to detect specific mutations in cell-free DNA. One can also use whole exome or whole genome sequencing to detect mutations in cell-free DNA. Data analysis and the interpretation, same thing. GATK best practice pipelines, ACMG guidelines, but there are different workflows and pipelines that each lab can use to address their needs. So the next one is a workflow for exosomes. Again, it's the same thing, but here it's not very clear in terms, especially of isolation. So ultracentrifugation, immunoaffinity, ultrafiltration, size exclusion chromatography. So it's very similar to CTC. So when we are talking about immunoaffinity, we are using surface markers again, like the same surface markers that are used for CTCs, and that limits the recovered amount of circulating tumor exosomes from plasma samples. Detection, one can detect using cancer-specific gene panels, RT-qPCR for RT-PCR or digital PCR, for DNA and microRNA. One can also use RNA-seq or microarray. For data analysis and interpretation, it's the same. It varies from the lab that's analyzing, but it's always usually based on GATK best practice pipeline and SCMG guidelines. So just to talk a little bit about the non-personalized versus personalized approaches for looking at somatic mutations in cell-free DNA. So the non-personalized approaches can be both in-house or through a service provider. And these include, for instance, like the Avenio ctDNA panel from Roche, or the Oncomine panel, cancer panel from Thermo Fisher, or also the TrueSight Oncology panel from Illumina. So one can acquire these panels and then use them in-house or use them in collaboration with either an academic partner, that they have established collaboration with. Or this can be done through Foundation One, for instance, or Guardian 260. So those, they are end-to-end commercial providers. You send them a sample and then they do all the sequencing data analysis using their existing technology. So the second option is personalized targeted assays. So for personalized targeted assays, I think this is the one that makes the most sense, but as I will mention later, it also has its own limitations. So the personalized targeted assays, so you start with your patient's tumor or the baseline plasma sample, and then you do whole exome sequencing, whole genome sequencing. Or you can use some large panels as well. And then you determine a list of somatic mutations from the tumor or from the baseline plasma. And then either you design a panel, an NGS panel, based on the somatic mutations that were detected in the primary tumor, and you use that panel for following disease progression or monitoring disease progression in these patients over time. Signatera assay is based on the same it's the same strategy. And also the other thing that you can do is to try to detect or quantify a set of these somatic mutations by digital PCR. So mutations that were detected in primary tumor can also be detected also using digital PCR, but I would usually look at 5 to 10 mutations at least to detect by digital PCR. So there is a limitation for this particular assay, for the personalized assay, is that the mutations that are detected in primary tumor may not be the same mutations 2 weeks or 3 weeks later when you are detecting in plasma and following up disease progression. That the drivers of that primary tumor may have changed, and then it's going to be an issue. That's why I said when you are doing, for instance, digital PCR, for instance, you need to do 5 to 10 mutations because some of them may not be there at that time. So to current challenges and limitations for circulating tumor DNA, circulating tumor cells and exosomes, I will try to go quick so we can get to the Q&A session. But for circulating tumor cells, again, the limitation is very low abundance, 1 to 10 CTCs per ml of blood, blood collection and stability, The issue that we discussed of epithelial to mesenchymal transition, also these are detected mostly in metastatic stage, high background of normal cells, low sensitivity and high blood volumes required. It's unclear what is the benefit of using CTCs in treatment decision making yet in clinical practice. For cell-free DNA, very low abundance as well, especially in early stage cancers. Blood collection instability, no standards to address pre-analytic, analytic, and post-analytic criteria. High risk of false positives, especially when we are getting at very low allele frequencies. And there's still some need for optimal methods for bioinformatic analysis. For exosomes, low abundance, high heterogeneity and nano-size, limited sensitivity and specificity, low purity and throughput, no standardized method. Active research is needed to develop an easy to operate, high sensitivity and high purity platform for exosome separation and detection. For the conclusions, obviously there are a lot of conclusions that can be made, but I just chose some. So obviously liquid biopsies have demonstrated promising activity in multiple clinical settings, but the great enthusiasm and expectations around them can be harmful if not adequately managed. Simple enumeration of CTCs like cell search system will not contribute significantly to the development of improved or more personalized cancer treatments. Technological advances in genetic sequencing of cell-free DNA had enabled liquid biopsies and led to marked increase in the therapeutically targetable mutations detection, as we have seen with the number of FDA-approved assays in cancer. The sensitivity of detection is improving with more novel NGS-based platforms, although these come with a higher cost and a longer turnaround time. The limits of detection have improved with new technologies such that the CAP-seq, the IDS, and SAFE-seq, allowing detection of variant allele frequencies lower than 0.01%. Prospective clinical trials are needed to explore whether changing therapy based on ctDNA dynamics prior to radiologic or clinical progression will improve outcomes compared to our current standard. Adoption of CTCs use or other non-ctDNA-based plasma biomarkers in routine clinical practice will require robust demonstration of analytical validity, clinical validity, and most importantly, of clinical utility. Because like I mentioned earlier, simple enumeration of CTCs will not contribute significantly to the development of improved and more personalized cancer treatments. So this concludes my presentation, and now back to Sonia.
Well, first of all, I just want to say thank you very much for that very detailed presentation. We are now going to begin the Q&A portion of the webinar. And for that, I would like to advise the audience that you can continue sending in your questions by using the questions window that's located on the right-hand side of your screen. And we'll try to address all your questions during this time that we have together. Now, I'm just going to pass the mic over to Mustafa to moderate the Q&A, and Lion and Christopher will join in with the answers. So you may go right ahead, Mustafa.
Thank you, Sonia. So I think the first question that we had was an operational question, and this is for Chris. So Chris, what are from a regulatory and protocol considerations, what are some of the considerations that need to be taken into account when enrolling patients into a clinical trial that uses cell-free DNA or ctDNA as a primary or secondary endpoint?
Yeah, thanks, Mustafa. There are a couple considerations, you know, when it comes to enrolling patients in clinical trials. A lot of protocols will have some requirements built into it surrounding certification of lab requirements. You know, you might see language around CAP/CLIA, ISO, IEC certification required. So I think it's important to, you know, as we're getting these results, that there's certification associated with those results. Regulators may ask at some point to provide that documentation, so it's important to track that across the life of the study. Along with that certification too, especially in Europe, with the implementation of IVDR requiring you know, results to come from CE marked labs or lab kits. I think that's an important consideration if you're enrolling patients into Europe, is to make sure CE marked kits results are coming from CE marked labs. Along too, even though some studies are allowing liquid biopsy results for enrollment, especially in like lung cancer and breast cancer, which Mustafa presented, we still may require biopsy, actual tissue biopsies as far as enrolling as well. So even though we might have that liquid component for eligibility, protocols still may require the actual formal tissue submission centrally.
And this is Ryan. One of the things I'll add to what Chris said is, one of the concerns we see, especially from FDA, is with percutaneous lung tissue biopsies. And in these advanced patients, whether it's a primary lung cancer or somebody with metastases, mets to the lung are so common. And the lung biopsy, if it's done percutaneously, carries a real risk of pneumothorax. And from a safety point of view, that's an FDA concern. So that's a strong reason that's pushed a lot of this push into the liquid biopsies. One of the earlier indications that got FDA approved was for the T790M mutation in lung cancer, to try to get away from the tissue biopsies for safety. So there is some compromise in mutation detection, but some potential improvement in not needing to have the patients with the lung primary or the lung met risking the metastases. And so much of the targeted medicine activity that's going on right now is in lung cancer, that it's making the liquid biopsies particularly important.
Thanks, Lion, and thanks, Chris. I just want to add also for, when designing a trial that use circulating tumor DNA, there are a number of other factors that can influence detection. For instance, surgery. So a surgery increases total cell-free DNA and can affect sensitivity of circulating tumor DNA detection. And the timing of circulating tumor DNA measurement must be taken into consideration, like 4 to 6 weeks post-surgery. There are, patients that are undergoing, for instance, active treatment with chemotherapy, that increases the total cell-free DNA and can affect sensitivity of circulating tumor DNA detection. So readouts from serial CT tests must be carefully timed to ensure reliable treatment monitoring. Patient weight also can increase total cell-free DNA and can affect sensitivity of circulating tumor DNA. Pregnancy increases total cell-free DNA and can affect sensitivity of circulating tumor DNA detection as well. And that may influence the initial enrollment into a trial because of the toxicities from treatment as well. Infections also can increase the cell-free DNA and can affect sensitivity of circulating tumor DNA. So all those are like considerations that need to be taken into account when enrolling patients for a clinical trial.
Yeah, and not so much for enrollment, but we do still see in a lot of the studies sponsors considering using the circulating tumor cell quantity as an endpoint. There's been a lot in the literature for specific tumor types. Prostate cancer is a very good example where there's been pretty good correlation between circulating tumor cell quantity and poor disease outcome. So not so much for an eligibility criteria, but for following patients over time, perhaps on drug, especially in a randomized study, to see if patients on drug and on the background therapy or a control arm are having higher levels of circulating tumor cells, and that in the future might be more of an endpoint. Right now, it's more of an exploratory or secondary endpoint, but something that's viewed in the clinical community as likely meaningful.
Okay. Yeah, thanks, Lion. So I think this question also is for you, Lion, about will tissue biopsy become obsolete in the future, or will liquid biopsy complement tissue biopsy?
So the tissue biopsy, when you can get the tissue, is still more or less the gold standard. That as well is far from perfect. We know there's tumor heterogeneity even within the tumors. And the primary and the met might have different profiles as well, and you can't biopsy everything and everywhere. But the tissue biopsy is still the ideal, but the liquid biopsy can avoid adverse events for the patient. And if the issue is really to find out, is a specific mutation present that might let somebody get a targeted agent or another specific therapy that they might benefit from only if certain markers are present, and a liquid biopsy is able to show that, that can have great benefit for the patients in terms of avoiding the invasive procedure. Because unless it's really a cutaneous metastasis, all biopsies are reasonably invasive. There's a big difference between doing a liver biopsy in terms of the toxic risk for the patient and doing a percutaneous lung biopsy. And sadly, as Mustafa pointed out, the glioblastomas don't do great at putting circulating tumor cells out to be able to be detected. And obviously the brain is not one of the easier places to biopsy, but for certain tumor types, the tissue biopsy is still going to really be needed. That doesn't necessarily mean for tumors that are metastatic to the brain that that holds, because there they've already metastasized to the brain, they were present in the blood. You've got advanced disease. That's a situation where there's more of a likelihood that the liquid biopsy might detect a tumor and therefore mutations meaningfully.
Okay, thanks, Lion. So I think another question that came up is, in addition to diagnosing cancer, guiding treatment decisions, and monitoring treatment resistance, what other applications might be on the horizon for liquid biopsy? So, Lion, do you think liquid biopsy may become, like the primary source of, let's say, for testing for MRD, for minimal residual disease?
It's coming along, and what's already sort of there in some of the tumor types in clinical usage already is the quantity of circulating tumor cells has already moved there. The liquid biopsying equaling, say, MRD like we have in chronic myelogenous leukemia, not there yet, but possibly in the future. If you take lung cancer is the best example of where, years ago we used to divide by adeno versus squamous. And now if you look at almost all the articles, it's the pie chart of, do you have any of the actionable mutations present? So whether that initial biopsy is a tissue biopsy or a liquid biopsy, if you can find those mutations and get the patients on a targeted agent that might have greater benefit for them and save the patient just being thrown onto a chemotherapeutic regimen with less of a chance of success and a lot more toxicity, that's the biggest benefit we've seen yet, is in that group. And that's then spreading out into all the other tumor types, but lung cancer is in the lead because of the large number of actionable mutations seen there.
Okay, thanks, Lion. Yeah, so another question that came up is, are there any promising liquid biopsy assays for early detection of cancer in asymptomatic patients? So as we saw before, it was more like late stage cancers, but I can take this question. So there are 2 blood-based early cancer detection tests that are under clinical trial investigation and maybe making their way onto the market soon, potentially changing the paradigm in cancer screening. And the results of which were actually presented during the last 2021 ASCO annual meeting. And they did show some intriguing finding in detecting early cancer in individuals. So this is early cancer in asymptomatic patients. So I think the first one came actually from it's the study of Galleri, which was developed by Grail. So the company has since been purchased by Illumina. So these have found that the test accurately detected cancer signals across more than 50 cancer types with an overall sensitivity of 51%. So that's a very good, actually a very promising one. The second one that they can think of is the early detection test that is in the Lunar 2 study, and then I think that one came from Guardant Health. So and that assay did incorporate ctDNA assessment of somatic mutations and tumor-derived methylation and fragmentomic, which is nucleic acid fragments across the genome patterns. So those are the 2 that I can think of that actually they are actively pursuing the approval, and they might be approved very, very soon. So I think we are getting at the top of the hour. I will, thank you again for all your questions, and I thank my, my fellow, Medpace panelists for their contribution, and I will go now back to Sonia for closing.
Okay, well, first of all, thank you very much, Christopher, Lyon, and Mustafa for answering all those questions. Yes, we have reached the end of the Q&A portion of the webinar, and if we could not attend to your questions, the team at Medpace will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that's on your screen, and that's info@medpacelab.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now give me a few seconds. I'm going to send you a link in your chat box, and you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Christopher, Lyon, and Mustafa, for that very informative presentation and for answering all your questions. So thank you very much, gentlemen. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone.